Okay, thank you very much. We are back, and in the previous video we talked about the cytokines involved in T cell functions, but now we're going to move on and we're going to talk a little bit about antigen recognition and co-stimulation. Okay, and continuing on, we must remember that the initiation of T cell responses requires multiple receptors on the T cells recognizing ligands on the antigen presenting cells. The TCR recognizes MHC-associated peptide antigens. CD4 or CD8 co-receptors recognize the MHC molecules. So what's important here to know is that the adhesion molecules, or integrin, strengthen the binding of T cells to the APCs, and the receptors for the co-stimulators recognize the second signals provided by the APCs. So what is it that recognize the ligands on the APCs and stabilize the binding of the T cells to the APCs? Well, of course, that is the adhesion molecules. So let's talk about these adhesion molecules. What is it that plays the important part in enhancing T cell responses to microbial antigens? Of course, it's the integrins. So let's talk a little bit more about these integrins. So how are these integrins used in the T cell response? What happens here? Well, first there is a weak binding of the antigen presenting cells by integrin mediated adhesion, and this is before the antigen recognition. And if the bound T cell fails to encounter the antigens, it disassociates from the APC. So just to summarize that, you can think that the T cell and the APC are meeting, and they are binding weakly using the integrin mediated adhesion, but if there is no antigen presentation, then they would then separate. So we have discussed that there is at first a weak binding with the integrin, and if there is a recognition of the antigen by the T cell to the APC, then there would be a strengthening of the bond. If that is, of course, if the APC is displaying the antigen that the T cell recognizes, then there would be an increase in the strength of the bond, and this would promote the stable adhesion of the T cell to the APC, resulting in T cell activation. How do naive T and B cells become effector cells? In order for this to happen, there needs to be two distinct extracellular cells. The first signal is provided by an antigen binding to the antigen receptor and is responsible for ensuring the specificity of the subsequent immune response. This means that there is a binding of the peptide MHC complex to the T cell receptor and to the CD4 or CD8 co-receptor. And then there is a second signal, and this is provided by the co-stimulators. So there would be B71, which is CD80, and B72, CD86 that are expressed on the professional APCs, which then bind to CD28 or CTLA4. Now we'll talk about it more later, but the CD28 on the T cell would promote the binding, whereas the CTLA4 would inhibit the binding. But let's go back a little bit and talk a little bit about the effector cells developing and talk about some basics. Now we can think about it quite simply that the first signal is simply a binding that happens because the T cell accepts the antigen. And remember that T cells are very specific for peptides, whereas the B cells are a lot more flexible in what they will accept. And anyway, in this case, this binding will happen, and the binding of peptide MHC complexes to the TCR happens, and also to the CD4 and CD8 co-receptor. So this is the first signal, quite simple, that there is an acceptance of the antigen by the T cell, by the presenting APC, and it accepts it and then moves on to the second stage. So again, to summarize, there is a first signal and a second signal. The first signal is an initial binding and accepting of the antigen, and the second signal is the complete activation and priming. So in the first signal, you would have the antigen presenting cell and the T cell. The antigen presenting cell would have the MHC, and the T cell would have the T cell receptor, and there would be an antigenic peptide which is presented and accepted. So this is partial activation or tolerance. That's the first signal. On the second signal, you would have the APC, and this would be a complete activation or priming. And what would happen here is that you would have on the APC CD80 or CD86, and on the T cell you'd have a CD28. So this would be complete activation and priming, and this would be the completion of the second signal. Again, to summarize, signal 1, and the antigen triggers the T cell receptor, and signal 2, co-stimulation is the critical second stimulation. So that should be quite easy to grasp. And there are a number of co-stimulators and integrins, and they can get quite confusing, but I'm just going to name a few, and then we're going to try to remember whether they are on the dendritic cell or the T cell. So how about CD80 or CD86? Of course, this is on the dendritic cell, and this would connect with the CD28 on the T cell. How about the MHC? This is on the dendritic cell. This would connect with the T cell receptor. This is where the antigen is passed over. How about the CD40 ligand? The CD40 ligand is on the T cell, and it would accept the CD40 from the, would connect with the CD40 from the dendritic cell. How about the ICAM1? This would be found on the dendritic cell, and this would